Again, thank you all very much for joining us today. I'd like to present to you a new option for cell culture. We like to call this presentation, "There's No Place Like Home." And I hope it will become evident as to why we use that title for this presentation. So this is primarily about cell-derived extracellular matrices, and what we're seeing is the next evolution in cell culture. So progressing to slide number 2, I want to start first by talking a little bit about cell culture, tissue regeneration, tissue engineering, and primarily just to note that when you think about tissues in the body, any tissue in the body is composed primarily of 3 different components, and that is cells, cytokines, growth factors, adhesion molecules, and the extracellular matrix. So those are the 3 primary components for any type of tissue. On the next slide, I like to show this image. This is an image that was given to me by Bob Meacham. This is a special technique called freeze etch or freeze fracture. So what you're seeing is a high magnification electron micrograph from scanning electron microscopy. And in the top of this image, what appears to be a smoother surface, that's actually a cell. That's a single cell. And all of the matrix, or the bird's nest that is on the lower part of this image, is the extracellular matrix. And this is it's a little bit different looking image because it is 3-dimensional. But I want you to focus in on the interface between that cell at the top of this image and the extracellular matrix. There is an incredible amount of information exchange going on between that extracellular matrix and that cell. And again, this is tissue. So this is the native environment for the cell prior to extracting it in most cases for cell culture. So just keep this in mind, the interaction between cell and extracellular matrix. So in this cartoon, again, standard cell culture methods usually start with tissue, which we're detecting or indicating here on the left side of the screen. We then pull these cells out or extract them from that extracellular matrix. And in most cases, the vast majority of cases, we put these cells onto polystyrene or cell culture treated plastic. This is a very foreign environment for these cells, and the first thing these cells are going to do is try to figure out where they are and how to respond to this new foreign environment. So what we're trying to do here is change that scenario in that you can isolate cells from their extracellular matrix and their normal environment and put them into an extracellular matrix that was produced and built by cells. So this is not a extracellular matrix that's been extracted or a protein that's been extracted, purified, and coated onto a dish. This extracellular matrix is actually built by cells onto the surface. And that's what we're using to put cells back onto when we do this cell culture method. Now, if we talk a little bit about the evolution of cell culture really didn't start until the 1940s. And it was in the 1940s that someone put a piece of tissue, intact tissue, into a jar that had support media. And that support media, they recognized that tissue they were able to maintain the viability of the tissue for some period of time sitting in that jar. So shortly after that, within 10 years, they decided, now I want to extract a particular cell out of that tissue so that I can examine it more closely and experiment with that cell type. So we started using cell culture dishes, and in this case, it's a glass petri dish. It was one of the first surfaces that was used, and in a lot of cases, they would coat that with a gelatin or something else to get the cells to adhere to that glass petri dish. Well, about 10 years after that, in the 1960s, polystyrene came along, and that pretty much screwed up everything. Polystyrene is very easy to use. It's very cheap. You can make it in a lot of different configurations. Most adherent cell lines attach readily to cell culture treated polystyrene, but it's easy. The problem is, as I mentioned before, it's a very foreign environment. Cells, in most cases, have not seen polystyrene before in the body. And so once you start a cell culture on standard polystyrene, it's a very different environment for those cells. So what we're trying to do is we're trying to change that. We're going back to the extracellular matrix and putting the extracellular matrix into a dish. As I mentioned, it's one of the 3 components of tissue tissues, but it's the one component when we go to cell culture that most people ignore. And that's unfortunate because it's very important. Understandable because it's not easy to do. It's not easy to make a substrate like this in its natural form. So what I'm going to want to do here is describe for you, and I'm going to show you a video just after this, which will do a much better job. Essentially what we do is we start with cells. These are cells that come from healthy human donors. We'll put those cells into culture. We'll expand their numbers to get a monolayer, induce them to lay down an extracellular matrix. And at the end of the process, we very gently remove those cells so that what's left is the extracellular matrix that they've built on the bottom of that dish. So it's not just the biochemistry, but it's also the structure, the way the cells built that extracellular matrix that we feel is important. So on this next slide, again, this is about a 2-minute video that describes the manufacturing process for our core technology, which we do starting with bone marrow mesenchymal stem cells that we purchase. And these are from healthy donors.
The following animation illustrates how StemBioSys takes human bone marrow stromal cells and creates CELLvo Matrix, a unique, natural bone marrow stem cell niche layered onto a standard tissue culture substrate. We begin by producing a working cell bank from a commercially available population of human bone marrow stromal cells obtained from healthy young individuals. These cells, along with a custom cell culture medium, are then added to a standard cell culture dish. The cells begin to divide, thereby increasing cell numbers and fully covering the surface of the cell culture dish or substrate. The cells are then stimulated to produce an extracellular matrix. These matrix proteins are deposited in a defined and ordered three-dimensional structure, a home built by the bone marrow cells in a fashion similar to what they do in their native bone marrow. This extracellular matrix is a complex, multi-protein composite that mimics the natural bone marrow niche for stem cells. Following production of their extracellular matrix, the remaining bone marrow stromal cells are removed very gently so as not to change the architecture or the biochemistry produced by the cells. The matrix is then thoroughly washed, the remaining solutions are removed, and the dishes or vessels dried, leaving on the surface of the cell culture substrate a native, three-dimensional home for stem cell proliferation similar to that from which they came.
So I hope that was much more clear in terms of the way that we process and build this extracellular matrix. And what I'm showing here on this particular slide in the background is, again, is a scanning electron micrograph of what that finished product looks like. So you can see that extracellular matrix that's been built by the cells. So the CELLvo Matrix, we have two different formulations. The original made with human bone marrow mesenchymal stem cells, and we use fetal bovine serum when we make that product, when we're initially culturing those cells. We now also have available a xeno-free version, and in this case, we've replaced the fetal bovine serum with human platelet lysate. So this is an all-human product, and I'll show you some data in the following slides that speaks to the performance of both of these products. So one of the things that we get asked quite often is, how are you sure going to different donors that you're getting the same product each time? And we've kind of taken the approach that there's over 100 different proteins present in these extracellular matrices. And so if you quantitate them or look at the quantity and quality of those matrix proteins, certainly the top 20 proteins are always the same from donor to donor. Once you get down to the numbers 90 and 100, they might switch quantitatively a little bit, but time and again, we're essentially seeing the same primary proteins that are present. So another way to look at this is on this slide here, we're looking at different donors that we've placed cells from each one of those donors onto tissue culture treated plastic or onto the CELLvo Matrix. And on the y-axis, you'll see cell doubling time. So obviously, we were excited to see that the cells on the CELLvo Matrix with the lower doubling time were proliferating much more rapidly in every donor that we looked at. But perhaps more importantly is the variation that you see with tissue culture plastic compared to the CELLvo Matrix. This is likely due, again, to putting these cells onto a natural environment or a matrix that they recognize. So we think these cells are just continuing to proliferate. They're waiting for a signal, which hopefully is going to be added by the researcher, as opposed to the cells reacting to their environment prior to experimental protocols taking place. So we're getting more rapid proliferation, and we're seeing more consistent results from donor to donor. So when we look at cell attachment, obviously this is an extracellular matrix, which is rather sticky. Cells adhere to it readily and start proliferating very rapidly. This is the same numbers of cells. These are cells isolated from human umbilical cord blood. Same numbers of cells put on tissue culture treated plastic for the CELLvo Matrix, and you can see the obvious difference in the amount of attachment. This is about 5 to 7 days after plating cells onto these different substrates. Here is looking at proliferation of different types of products. Starting on the left, that's tissue culture treated plastic. Then we have our two CELLvo Matrix products, the standard product and the xeno-free product. Then we have coated dishes. So this is a fibronectin coated dish, a collagen 1 coated dish. These are made by BioCoat type dishes. And then we have two different competitors' products. These are products that are used specifically for mesenchymal stem cells. And I should mention the cell type here is also a bone marrow mesenchymal stem cell that we put onto these different substrates. And you can see the significant difference in the numbers of cells when they're grown on our CELLvo Matrix products than on these competitive products. We see the same results, certainly with mesenchymal stem cells of different types. So the one we're showing here, I believe this is from adipose-derived mesenchymal stem cells, but we see it with bone marrow, adipose, umbilical cord blood, umbilical cord tissue. Different types of MSCs respond the same to this extracellular matrix. It's been our experience. Looking at some different indices, this is actually some work that was done by Robert Langer's lab at MIT. This is a separate study from what I had shown before. They were looking at competitive products again, and this is a bit small, but you can see that we're getting more cells, so they're proliferating more rapidly. The cells are smaller in size when they're on the CELLvo Matrix, which is an index of MSC potency. And then one of the markers that we've keyed in on, essentially from the onset, our scientific founder had an interest and was always looking at stage-specific embryonic antigen 4, or SSEA4. And it's actually part of our QA/QC criteria when we build each lot of this material as we look at SSEA4 expression. I want to see a significant increase in the number of cells that are expressing this potency marker. So this is an index of, again, a potency of those cells. Again, the comparison to tissue culture treated plastic in the CELLvo Matrix. We also see this as an index of responsiveness of the cells to growth factors. And there are publications from our scientific founders lab specifically looking at bone and bone morphometric protein. We see that cells that have been grown in this matrix are much more responsive to growth factors than they are when they're on plastic. And this is likely an index of those cells being in a foreign environment and trying to lay down an extracellular matrix, while when you put the cells on a natural substrate, not only are there places in the extracellular matrix to express and present growth factors and cytokines to the cells, it's just more effective, or we found it to be more effective in several instances. And this is a heat map for gene expression. Again, more work done in Langer's lab at MIT. And this, we won't go into detail of all the different genes that are being expressed, but this is just to show, again, in a controlled experiment, same numbers of cells. Again, these are bone marrow MSCs in this case, put onto the CELLvo Matrix or put onto plastic. You see a very different gene expression profile. So perhaps it's obvious, again, going back to the cells are responding to their microenvironment, and they're going to express different genes based on the feedback that they're getting from that microenvironment. And that's what we see here. So we've also found, over the period of time that we've had this product available, that it's not just mesenchymal stem cells that respond favorably to this extracellular matrix. So the mesenchymal stem cells that I've mentioned before, certainly, we've also found that chondrocytes, endothelial cells, cardiomyocytes, human islet cells, cancer cells, different types of germ cells, in this case oocytes, and different types of bone cells, all have shown increased proliferation and responsiveness on this extracellular matrix compared to other substrates. So it's possible that the matrix being produced by a mesenchymal stem cell is able to support the growth of different types of cells. And that's been our experience as well. Just a quick look at some of those competitive products that I had mentioned before. BioCoat can be used for different types of cells. CellStart and Mesencult products were developed primarily for mesenchymal stem cell growth, and those are some of the comparisons that we had seen before. And then we hear a lot about Matrigel. We're actually have a product development project going on here to develop a cell-derived extracellular matrix that performs like Matrigel. We have not seen with this particular product, the bone marrow product, that it's especially good at supporting iPS cell growth, just as Matrigel is not good at supporting the growth of mesenchymal stem cells. So going back to the comparison again, tissue culture treated plastic, again, the fibronectin and collagen, these were BioCoat products. Competitor 1 in this case was the CellStart product, and competitor 2 was the Mesencult product. So what we've also done now going forward is we've decided that we were going to provide these more potent cells to researchers as well. So we've taken on an effort to isolate these different types of cells that you see here, the adipose MSC, the cord blood, endothelial progenitor cell, chondrocytes, bone marrow MSCs, and wart and jelly MSCs from the cord tissue. And these cells are different from normal MSCs that you see in other catalogs in that they've been isolated and grown on this extracellular matrix. So the gene expression profile, the proliferation rate, and the other things that we've shown you are going to be different on these cells that you get from StemBioSys and on different distributors than mesenchymal stem cells that you'll see on the market from others that have been grown primarily on tissue culture treated plastic. All of these cells, with exception of the adipose MSC, are now available. So the CELLvo difference, why the matrix? As I've tried to describe, it provides a natural substrate for cell isolation and growth that cells are responding to. It allows these cells to be more responsive to experimental variables than to a foreign substrate or microenvironment that they might find themselves in during normal cell culture processes. Other substrates only contain one or a few extracellular matrix proteins, which is very different than their native environment. And one of the other things about this matrix, obviously, is it's ready to use. It's on the dish. We ask that you add your cell culture media or just PBS to these dishes for a period of time just before you start your cell culture. There's no coating required or overnight processes. It's a very unique product. There are no other products like this out on the market that have a cell-derived extracellular matrix. And the cells are unique in the way that I just described. They've been isolated and grown on the CELLvo Matrix products, and therefore they're exhibiting a more natural cell phenotype. So we have the matrix products. We now have cell products, and we plan later this year to be coming out with our own CELLvo media. This will be a basal media. We'll have a xeno-free formulation and one with fetal bovine serum. And as we go forward, we'll probably be bringing out media products specific to the different cell types that we offer as well. But that's part of our pipeline coming up in the near future. So I'm going to end here just with a slide that shows our different distributors. As I mentioned, Caltag Med Systems in Europe, in the US and Canada. Obviously, you can go to StemBioSys, but PWR is our primary distributor here in the US and Canada. In Japan, we have Funikoshi, and in South Korea, Solin Bioscience. Again, thank you very much for joining us today. We've enjoyed speaking to you about our products.